So thank you very much. I was excited about this talk when it was originally at KOP, which is like 20 minutes from my house. Now it's even closer. So just as a overview, so my background is I have a PhD in mammalian genetics. I did a postdoc at Pfizer and at Yale in diabetes work, and that launched me into years and years of working in drug discovery at ISIS, now I called Ionis, and then at J&J. And the basic work that I've always achieved or tried to achieve is trying to partition fat out of the liver and the muscle through energy expenditure. So this built up my expertise in mitochondrial research, which eventually I left J&J to try to start this company, Mitochon, and luckily in 14, we got the study. The company launched with Ben Franklin Technology Partners and Robert Alonso. So basically, you can see that the focus is really on really insidious diseases. There's no work done whatsoever in diabetes, even though that's my expertise. And so today I'm just planning on trying to hit across the treetops and give you some idea of what we're doing, how this platform works, and where we're going. So there's usually I end with this slide, but I'm going to start with this. And basically the question is like, why are so many indications require aging? And it's likely for ALS, MS, Huntington's, Parkinson's, all cancers, that you need a decrease in repair over time, and you have probably an increase in mutations, mitochondrial dysfunction, and then bam, you have all the things necessary to see onset. So, but if we had a way to slow down repair and increase mitochondrial functions, potentially through endorphins, brain-derived neurotrophic factors and things like that, then potentially in the pre-manifest stage, you could start dosing and you push off all the manifestations, maybe for either forever or for a long time. So this is my little cartoon where, I worked in drug discovery, and we always have these dogmas and these ideas, and we work on them forever. And then we find out, that it's completely wrong at the very beginning. So here's this guy, colleague on the left saying, here's your mistake, which is at the beginning. And depending on how long this takes for you to catch up, it can be 5 to 20 years, and it can be millions to billions of dollars. And one of the things that I think fall into this area is probably work that was done in Alzheimer's. So this is a paper that came out by Virginia Lee, where she aged Alzheimer's mice for 18 months. And every single month, she would do a measurement of what's called isoprostanes. And isoprostanes are a lipid that gets attacked by a free radical, hydrogen peroxides or superoxides, and put a double bond in it, and it shows up in urine, blood, cerebrospinal fluid. So in this model here, she shows that at 8 months of age, you have isoprostanes coming up, but it took you another 4 months to see the first signs of plaques. So we always think that when it comes to Alzheimer's, that the plaques is a downstream event, and mitochondrial dysfunction is upstream. And if you look at isoprostanes, which is our biomarker of mitochondrial reactive oxygen species, they're higher in Alzheimer's patients than non-Alzheimer's patients. This is urine. You can also see it in Huntington's disease patients. They're higher in the cerebrospinal fluid versus controls. People who have multiple sclerosis, no matter which category you're in, but in particular in the transition from early to late, which is called secondary progressive MS, very high isoprostanes, but it's still high at relapse and remitting and primary progressive. And then people children who have Duchenne's and adults who have Becker, very high isoprostanes. So all these diseases appear to have a mitochondrial dysfunction to it. Children with autism have extremely high levels of isoprostanes, and it doesn't matter where they fit into the bucket list, they're high. So there's a paper that came out many years ago. Martin Brand is one of our advisors that says that mitochondrial reactive oxygen species production is very sensitive to mild uncoupling, mitochondrial uncouplers. And so this is the platform we work on. We work on it's likely, so you guys were talking about CACO2. This is a molecule that's likely a class 1 molecule, and that it doesn't require any transporters to get on board. It's brain penetrant. The molecule that we work with is called 2,4-dinitrophenol, used in the 1930s for obesity, huge doses, and we use it now at hundreds of fold lower doses. Has a dissociable proton, and it has two electron withdrawing loops groups, the nitrogens, that pull the electrons away from the hydrogen, and it becomes weakly held on. Out of all the organelles inside your cell, the mitochondria is the only one with a pH basic environment. So one-to-one chemistry say that a weak acid like dinitrophenol, acids go to base. So it specifically travels to the mitochondria, and this is where it creates what's called the non-genomic effect. The target is the mitochondria. It's not an enzyme, not a receptor, not DNA. And it releases the proton into the pH basic environment of the mitochondrial matrix. And then comes out as an anion, gets reprotonated in the acidic environment, and goes back in to drop off another proton. And this is actually a natural occurring effect called proton leak, where you lose 30% of your energy to protons leaking across the membrane, and thought to be evolutionarily a way to slow aging. But it does increase energy expenditure, and this is the reasons why it was used for obesity. So here's the non-genomic effect. So the mechanism is based on biophysics. The molecule travels to the mitochondrial matrix. Right here is, we call it MP101, or is our 2,4-dinitrophenol. It goes in and drops off the proton. This changes the mitochondrial membrane potential, which immediately prevents the overt production of reactive oxygen species. And it then closes the uniporter for calcium influx. So you have a lowering of mitochondrial calcium, an increase in the cytosol, which we think activates the adenylate cyclase. It does increase cyclic AMP, and then cyclic AMP activates CREB, and CREB is a transcription factor for BDNF, which a lot of people consider a fertilizer of the brain, involved in cognition. Eric Kandel's cognitive factors are cyclic AMP, CREB, BDNF. So the non-genomic effect on the left eventually cascades into a huge remodeling of the cell, and even in the mitochondrial population. We've put the molecule into wild-type mice for a couple of weeks and showed that there's a nice induction in the cortex of BDNF, brain-derived neurotrophic factor. And so this is a protein that is involved in bathing of neurons, and it's also a myokine outside of the brain in muscles. Actually, it's anti-diabetic as well. But there's a law of diminishing return for how it works, in that the lower doses appear to be better than the higher doses, and we call this a hormetic effect. So one of the first studies we did is in a genetic model. So DNP has been put into wild-type mice and rats, but it's never really been put into a germline mutation where it's very, very fragile mouse model. And then for this, we picked the Huntington's disease, which it's an autosomal dominant mutation in humans. You get it around 40 years old to 50 years old, and depending on the expansion of the CAG repeats, basically dictates how aggressive it is and how early you get it. We use a model called the N17182Q, which is human transgenic gene that has the pre-manifest stages similar to Huntington's disease. Work was done at Johns Hopkins. So 17 weeks with DNP by oral gavage every day, including weekends. And the typical dose window here is 0.515, which is basically an equivalent dose in humans to 2.5 milligrams to 22 milligrams. Compare that to the 1930s where they gave it to people at 300 milligrams. So 3 milligrams versus 300 milligrams, completely different. So we we did MRI scans to show that in the mice, their brain volume was significantly preserved. And one of the things that you lose with Huntington's is your brain volume, brain mass. And so to understand why the brain mass may have been preserved, two cell types were looked at, spiny neurons and general neurons. So this is a biomarker called DARP32 of spiny neurons, which are the ones that are specifically killed in Huntington's disease patients. And then PSD95, which is a general neuron. So we have the work, we did this in cortex and striatum, and basically here's the 1 MPK dose of DNP after 17 weeks, which is looks equivalent to wild type, but the placebo is significantly reduced. And then even though it's historically a weight loss drug, it's completely weight neutral at these doses. Wild type animals are bigger. Here's the 5 MPK dose of DNP, didn't change the body weight here in the wild type. And all the doses here in the Huntington's mice are basically weight neutral. So it's apples and oranges. And then the big deal for us is the isoprostane. So we took the cortex and sent it off to Vanderbilt and showed that the isoprostanes in the 1 MPK group is basically equivalent to wild type, but the isoprostanes are higher in the placebo HD mutant. So this is, again, a biomarker of reactive oxygen species. So switching into a different model, we then tested it in Alzheimer's disease. And Alzheimer's represents 73% of all dementia. And in this case here, we did use the APP-PS1 mouse. This was done at Mark Matson's lab at the NIA. We aged the mice for 4 months and then dosed the mice for 4 months. And at 8 months of age, they were evaluated in the Morris water maze, which is where you submerge a platform and they have to find the platform underwater. This is at 8 months of age, this is the training of finding the hidden platform. So it's the lowest dose here, which is 0.5 MPK, around 3 milligrams per day for humans. The mice learned faster. The drug accelerated learning in a sense. And then on the day when the hidden platform was removed, the animals at the 0.5 group in particularly were obsessed on where the platform used to be. So it keeps crisscrossing, entering that same little space, which is about the size of a business card, and because they knew it was there. And this was significant. Well, it went out to 72 days, 72 hours. So that was a hallmark of, short-term memory is a hallmark of Alzheimer's. And then to improve upon the wheel, DNP is fine, but we wanted to get a new molecule made. Using DNP, we made a prodrug. This has an extended PK profile, longer half-life, lower Cmax, enhanced pharmacology, and also it has a new chemical entity. So we have patents on this one. So for here, we took both molecules for a walk in multiple sclerosis. This is completely different than Alzheimer's and Huntington's. And here, there's no known cause. It's an autoimmune disease. Antibodies attack the myelin sheaths in your brain, and also, if it progresses, it'll attack the axons. High levels of reactive oxygen species, as I showed before. And it disrupts voluntary movement and involuntary control of bladder control. So here we use the prodrug and the DNP, MP101 Prodrug MP201. So here's the most basic model. It's called the EAE model, where they provide a peptide of myelin, which starts the induction of the autoimmune response. Then out at day 7, we start treating with our molecules. Peak is usually at around day 15. What you're looking at here is someone going into the vivarium every single day and measuring the degree of paralysis. So 5 is death, here is severe paralysis. And you can see that in all the dose groups here, that low doses of an uncoupler have lessened the degree of paralysis. And then at 2 weeks, it stops and they recover. And here's an interesting thing about the drug is that it actually prevents wasting. So all the groups here, DNP and the prodrug, have preserved body weight, whereas the placebo is actually wasting away. And it's also one of the things we think about is applying the drug to severe burn victims, are usually the most sick people in a hospital, to prevent cachexia. And here's a little video to show you that the drug is really disease modifying. The first is going to be the placebo, where the mouse is paralyzed at the bottom of the cage. And then here is the 5 mpk of DNP. There's no paralysis. And then the prodrug at the mid dose, and then finally you'll see the highest dose. And this mouse actually hops out of the cage. So it's really a strong effect. And then related to the mitochondrial population, I mentioned that it changes the population. So here is the mitochondria from untreated EAE mice, and they are shorter. And they also have this weird Y-shaped mitochondria here. But after dosing for a week, population got shifted at 5 mpk of DNP and the prodrug back to the normal length, and the Y-shaped morphology went away. So we call this the culling of the mitochondria, or mitophagy. And then the other interesting thing is that here is the Western blot from the spinal cord and PCR. So here is DNP and the prodrug relative to the vehicle, and myelin basic protein has been preserved, which is one of the things that gets destroyed in this model. Markers of damage are reduced relative to the vehicle. Here is BDNF, which is our neurotropin. And then the family of inflammatory cytokines, IL-1, TNF, INOS, are also all reduced. So it has this pleiotropic effect of affecting many different pathways. And then the most recent paper is the Parkinson's work we've done. We've also worked with Mark Mattson on this. The previous slide was work that was done in Japan on MS, Yoshi Bando. This is published. So this is also a no known cause. And for this particular disease, we use the 6-hydroxydopamine injected into the right striatum. And then of both low doses of the DNP and the prodrug, dosed for 2 weeks, and 6-hydroxydopamine over the 2 weeks is going to destroy the dopaminergic neurons. At the same time, we're trying to protect them from being destroyed. So this is basically happening in synergy, or simultaneously. And so the thing that happened with a nice effect here is that here's the wild type controls, and here is the low dose of DNP and low dose of the prodrug compared to the vehicle. And the vehicle lost the dopaminergic neurons severely, and at both DNP and the prodrug, this population has been protected. So I mentioned reactive oxygen species. Isoprostings are a huge issue for many diseases. The other issue that probably is less appreciated is something called calcium overload. And so for children that have Duchenne's, or people who have ataxia, Huntington's, epilepsy, Wolfram syndrome is one of my, these are all associated with overt mitochondrial calcium overload. And if you don't get calcium overload under control, then it'll lead to apoptosis. And there's a recent researcher that we're working with, Lon Lepere at Rochester, who has this hypothesis about ALS, and that ALS is caused by the calcium overload at both the muscle cell and the motor neurons that cause the reason for the neuron to lift off the muscle, which cascades into the atrophy of the muscle, Lou Gehrig's disease, or ALS. And I told you earlier that one of the things that DNP does is close the uniporter. So here is the intramitochondrial calcium levels. They're lower with DNP. And so this might explain why we had such great pharmacology in a model of Duchenne's muscular dystrophy. So Lee Sweeney dosed this model called the MDX mouse with DNP for 3 solid months, and then measured the diaphragm strength. So here's the data where it shows the 1 MPK and 5 MPK of DNP in this MDX mice. Here's where the diaphragm has been taken out, put between 2 holders, and then they activate the diaphragm to pull. And out of all the drugs he said that they've ever tested before, they've never seen a drug that has this high diaphragm strength. And then here's prednisone, which is the gold standard. DNP at both doses were better at diaphragm strength than prednisone. And then in a combination with prednisone and DNP, DNP did not worsen the pharmacology of prednisone. So basically the idea was that to see whether or not it would cause any harm, and it did not. So the calcium overload also is probably the reason why we have a nice effect in traumatic brain injury. Traumatic brain injury is associated with a lowering of respiratory rates, high amount of losses, and calcium overload. So in this study here, cortical impact, which is like a little BB hit in the cerebellum, was used to create the trauma. And then dosing began at 2 hours post-trauma to sort of like mimic the human scenario. And then the results is sparing of the tissue, what I call the threatened tissue, and improve cognition. So here's the vehicle. It's basically, you can see the cerebellum. It's got a sort of like a bite out of the apple. And with the prodrug here, we have a little nibble of the apple. And then 38% preservation of mass that would have been lost, which translated not the first week, but the second week into improvements in novel object recognition. So this is, you preserve the tissue and then you want to get the cognition back. And so this shows you that the mice that are treated with the prodrug got their curiosity back. And that finally brings me into ALS. So ALS is also a motor neuron loss. It's 90% are known to be sporadic, severe wasting syndrome. There's no disease-modifying drugs to date, and so we got luck and have this great researcher, Lon Lepere at Rochester, who took this on. So she dosed the SOD1 mice, which is the gold standard mouse for ALS. And even though it's sporadic, this is the one that they are typically used at a dose of 0.5 MPK. And I mentioned again, this is around a dose in humans with 2 mg per kg. So this is, the study's ongoing, but basically this is the rotor rod they have to stay on for 60 seconds. So everyone does a great job staying on the rotor rod for 60 seconds at 5 revolutions per minute. This is wild type. Then the vehicle of SOD1, and then our DNP MP101 dose group. But when it gets up to around 30 revolutions per minute, the placebos are falling off. But both the prodrug and the prodrug DNP seems to be almost equivalent to wild type in being able to stay on. So this is, we're building up the N on this study, and there's more data than I have here, but it is pretty impressive that such a low dose over a long period of time has preservation. So the thing about DNP is that it was used for obesity in the 1930s, but it was never approved as a drug. But the FDA granted us an open IND, and we've started phase 1 this year. Right now, we got through the first cohort, and then Corona came in and put us on hold to get to the next dose. But the PK and it was great, and there's no adverse events. So we're pretty excited. So the goal is to finish off phase 1 in normal healthy volunteers this year, and with the idea of potentially, hopefully, getting into phase 2 in a series of studies next year in Huntington's, ALS, and Alzheimer's patients. And just to put this in perspective for regular, I guess, more pharmacology of drugs that you're probably used to, this has an advantage in that since the target is the mitochondria, and the mechanism of action is based on biophysics, and the mitochondria are extremely well conserved among mammals, there's a good potential that translation is superior versus going after a receptor or an enzyme from mouse to human. So the basic gist of what we want to do is to enroll patients who have high amount of reactive oxygen species, isoprostans, in the cerebrospinal fluid. And then then enroll them specifically for this, and then dose them for 14 days, then take the cerebrospinal fluid again at day 15 and look at the delta for key biomarkers. So a consortium of ROSs, antioxidants, markers of inflammation, basically borrowing from many different studies on what the pharmacology should do. And then we would specifically have 3 different studies: a study for ALS, a study for Alzheimer's, and a study for Huntington's. And it should help us to do dose finding and, of course, demonstrate target engagement. And then we would circle back to do the critical study, 1 year plus studies. So there's collaborators. We have a lot of collaborators: Mark Mattson, Michael J. Fox, Wenzhen did the work on Huntington's, Yosh in Japan, ALS's work going on in Rochester. Lee did the DMD work. Patrick Sullivan has the TBI work, and we just got a R01 grant. Well, he did for looking at TBI further with the prodrug that's worth $3.2 million. And that work will go on at University of Kentucky and here in Philadelphia at CHOP. And I mentioned the original seed money came from Penn Frank Technologies, which was a great way to get us launched. That is everything.
So when you did you expect activation of mitophagy as a consequence of uncoupling mitochondria? But you would expect activation of mitophagy, which seems to be also affect a defect correlated with progression of neurodegenerative diseases, correct?
Well, so I have a colleague, his name is Orion Sharif, and he focuses on mitophagy. And so basically the idea is that mitochondria that are dysfunctional, poor mitochondria, they produce less ATP and they produce more ROSs. They typically have a lower membrane potential. So this population, by luck, is the population that we can easily target by lowering the membrane potential even more, which sends them into mitophagy. So I think the healthier ones that you have, so I always look at mitochondria as a population. It's relatively mosaic in a sense. You have good ones, bad ones, short ones, long ones. So I didn't initially expect that it would do that, but and I didn't really know that it would actually have the induction of BDNF either. My original idea for the drug was to lower reactive oxygen species. And according to experts in the field of ALS, Parkinson's, Alzheimer's, they said that if that's all you did was lower reactive oxygen species for Alzheimer's, Parkinson's, then that would be disease modifying. So that's how originally we started. And then it ballooned into something much bigger: mitophagy, BDNF, and calcium overload. So you can see that it has a broad neuroprotective effect by modulating the entire mitochondrial physiology. I can't hear you.
For example, there's a correlation with defect in mitophagy, cell senescence, increased inflammation, and then there's a relationship in terms of mitophagy, lack of mitophagy in ischemic injury, neurodegenerative disease, Parkinson, Huntington disease, Alzheimer's disease. So in terms of the link ultimately between reactive oxygen generation and activation of mitophagy, for example, is well established. So the question comes really in terms of when you look at mitochondrial health, but there's multiple entry points in a way which all end up in a way of restoring the health of mitochondria. So the question is in terms of focusing on mitochondrial uncoupling, in terms of is there must be other strategies there, okay, which ultimately in terms of working in the synergy in terms. So anyway, it's just a more philosophical questions at the moment. I wish you good luck with them as you talk discovery, by the way.
Right. So I think it's mitophagy that creates the hormetic effect. So I look at it like the dose is almost like velocity of turnover. And it's possible that the reason why we get such great effect at low doses, and maybe higher doses are required for other indications, but I think it's the low doses that cull the bad mitochondria and replenish it with a better population of better APT producers, lower reactive oxygen species producers. But then if you push the dose higher, then you're starting to get rid of the good ones now too, maybe. I don't really actually know, but this is an area that we want to eventually explore is why is that, what's the exact basis of hormetic effect? Kind of titrate out things as well between the ROSs, the calcium, BDNF. Everyone wants it, people like a nice clean one drug, one effect kind of thing, but this is not it. But for the good news for anti-aging, it's working on really insidious diseases. And I think these diseases need these multiple pathways to be modulated to make an effective drug. And in a sense, it's a low-hanging fruit to by waking up compensatory mechanisms with an oral small molecule that's brain penetrant before you get into more heroic things like gene therapy. If I can wake up compensatory mechanisms with an oral small molecule, and that pushes off manifestations of the disease, then that is really a low-hanging fruit.
Can I ask a quick question, please?
Yes.
Sure. Yeah, so great talk, really interesting looking compound. I wonder if you had any thoughts coming back to animal models, especially for neurodegenerative diseases, not necessarily translating well for patients, because we've had a lot of drugs that look pretty good in an ALS mouse that hasn't translated, sure, including things like alizoxime, right, that was proposed to work around mitochondrial function. Do you have any thoughts on some of these other agents that didn't translate to patients that may have been working on the mitochondria?
Yeah, so I think there's a couple of things. One is that most of these drugs are perimitochondrial. They're not actually mitochondrial. This is a drug that actually specifically modulates the mitochondria. That the target is the mitochondria. So if you're working on antioxidants, you're sort of perimitochondrial. You're mopping up reactive oxygen species after they've been made, mito-Q, co-Q. It's too late. So the better way is to prevent them from being made in the first place. The size of the molecule that are used in the past, sometimes some of them might be not the best brain penetrant, tissue penetrant. And then, it gets into the cascade of having the full pharmacology here. So it isn't just reactive oxygen species it does. It doesn't just do calcium, but also induces BDNF, Krebs, cyclic P. So in that Milo, I think is where you really start to get this, get the pharmacology. And it wasn't, something that I knew initially. Initially, we started off with Huntington's and a very, very fragile mouse model. And, the drug could have killed everything. And then it would have been a dumb idea, right? But so I was really on pins and needles for that time. And luckily it preserved brain volume, preserved motor skills, which I didn't show that data, but it's published. And that started us down this road where it ballooned into something, the next indication. So we're basically 10 indications in. and somewhere batting 1,000. Rett syndrome, hearing loss models. But I think that, and I'm an in vivo pharmacologist, so I only want so much from my mouse models, and then I don't want to go back. I want to go sort it out in humans. So that's the idea right now, is that a lot of people get duped by preclinical models, the SOD1. I could easily fail in the SOD1 and be 100% confident that I could be successful in a human, even if it failed in SOD1.
Definitely. Yeah, because there's been a lot of things that have worked in, well, not a lot of things, but there's certainly been things that have worked in SOD1 that, haven't translated at all to patients.
But I think also is that now that we used to be a criticism that people said is like, oh, you got the magic elixir, that does it all, and which, is kind of insulting, but the drug modulates the entire mitochondrial physiology. And if it does that by changing membrane potential, CREB, cyclic KMP, everything, then you could have a drug that has this broad protective effect. And so I think that the fact that it's worked in so many different indications by completely different people working on it, and gives me creature comforts that it's real.
Hope so.
Could I just make one comment regarding the therapeutic efficacy or the therapeutic index? One of the things that you pointed out was that at high doses you lose the effect, but at the lower doses you seem to have a good effect. The reactive oxygen species actually exhibit auto-accelerating and decelerating effects on mitochondrial function, in that, for example, if we look at a gain of function in SOD, if you look at trisomy 21, there's 30 to 50% more SOD function, and yet those individuals with Down syndrome have accelerated development of dementia and Alzheimer's disease. And so scavenging too many of the oxygen radicals may actually impair the mitochondria function. And so your drug at the lower doses is probably sopping up the reactive oxygen species that, and as you say, culling those mitochondria. Whereas at the higher doses, you may actually lose the effect. And so the pharmacokinetics, or the amount of drug that you deliver to your target tissues, may be critical in optimizing the dose. And you might have to tailor it to individual patients.
I don't know if necessarily to individual patients, but I do think that it's, I think for each indication, there's probably a tweaking that needs to be done. Like it looks like for me, for MS, they like a little bit higher dose. Parkinson's and Alzheimer's and ALS, it's a really low dose that seems to have we haven't sure, it seems to have the best effect. So, but I also think there's BDNF is also in there. So there's this window where even the lowest dose of BDNF was trending to be positive in the cortex in that 2-week study. So now we're talking about 17-week studies and 3-month studies and stuff like that. Now you have plenty of time for BDNF to come up. And BDNF also is, I mentioned, is a myokine. So the bathing of a myotube with BDNF also has some effect, even though it's, everyone's always focuses on BDNF and the cognitive factors. So I think that in general, in most pharmacology studies, if you don't have an effect, you double the dose, right? You go to the higher dose. Well, in this case here, it might be that we actually need to go down.
Yes, I would agree with you on that. Yeah.
But we're going to start off with the doses right now intended are probably for all these indications are somewhere between 1 to maybe 10 milligrams per day for human. In the 1930s, they used 300 milligrams to even more, and currently, weightlifters take the drug and they don't take anything below 200, I'm told, because it doesn't do anything. But in that 1 to 10 milligram range, it does everything. That no one wants to lose their mind.
Mhm.
So, but it won't do anything for your muscles. Actually, they use it for fat. They're trying to burn off the last bits of fat. So they take a big dose, short duration of time, which is not our program at all.
Right.
I have one question regarding other cells that are going to be targeted by this drug, because it is not going to be only neurons. You can imagine that immune cells and whole body is going to be targeted by this. Mm-hmm. How, especially in long term, what would be your expectation? Long-term usage of this drug in low dose, how would you envision how it is going to happen? What's going to happen?
Yeah, so it is true that it basically penetrates all cells, all tissues, all cells. I mean, it's likely a class 1 molecule with no transporters. And we've done tissue levels, and in the brain, muscle, liver, they're almost the same. So it isn't like they don't only, your liver gets the most, and then you get like 1% going into the brain. That's not the case here. It really is a brain penetrant molecule. Anyway, so to get to your question is that somebody's actually done this study. They took a low dose of DMP, gave it to an entire mouse colony for their entire lifespan. And it lowered the amount of hydrogen peroxide that was produced in the brain, liver, muscle, heart. It lowered the amount of DNA damage. They lowered the amount of oxidized proteins in the liver, brain, heart. It lowered circulating insulin, glucose, triglycerides. And the consequence was the mice lived longer than water. So I've enrolled this molecule in, it's called the Intervention Testing Program, which is one of the NIH's programs to find a drug that can potentially push off all age-required diseases. So this actually just started, I think, this month, despite COVID, and you'll have to wait about 4 years to find the result. But it is probably an anti-aging drug with lowering ROSs and increasing BDNF and other things, but a very low dose, long duration of time.
Okay, thank you.
Since fighting infection, hydrogen peroxide bursts are important essentially to get rid of your infectious agents. Would that affect essentially your defense mechanism by reducing the production of reactive oxygens? Could that be a potential side effect?
We've done the tox studies, and we've done the rat dog tox studies, and nothing's popped up that's an issue, and we're going to go back to do the 6-month rat, 9-month dog, because our IND right now is open for 14 days. So what happens in long term, I don't know. But I can tell you that the molecule didn't have any off toxicity. It was pretty clean in the rats and dogs.
I'm not necessarily talking about toxicity. I mean, it's more or less essentially reactive oxygen regulation that plays a role in so many different physiological processes there, and it's constantly fluctuating. It's a dynamic process, the regulation. And one of the components there is when you get inflammation and infections, that you have essentially macrophages. You get this oxygen burst in a way, which is part of the defense strategy. So the question is, would you expect that type of defensive reactive oxygen bursts will be impaired, and could that have consequences? I'm speculating now here.
Yeah, so, I don't know. In the MS model, of course, it lowered TNF alpha, it lowered INOS, it lowered IL-1 beta. So these are the inflammatory cytokines in the storm that you see. And same thing I would expect in Duchenne's. Duchenne's has that inflammatory storm that takes place early. So I would suspect that the pharmacology would translate into other indications, but I don't really know. I mean, I don't know.
Yeah.
I mean, so far it looks like it's working.
No question. We're going to see an effect. No question.
Yeah. So I mean, I don't. I'm sure there's indications where it could be worse. Maybe some kind of lysosomal trafficking or some kind of thing where you don't want to accelerate these pathways. There's so many indications. But in the end of the day, Martin Brand, my great guru in mitochondria, he just says, every time I talk to him about, what do you think about, would it work in this, would it work in that? And he says, John, for something like this, the only way you're going to know is to test it. you can sit there as an armchair scientist, think about it forever and why it wouldn't work, you know, and then in the end of the day, you really just have to test it. And
that's the way how discovery are made. Good luck. Okay.
Yeah, so I think what's interesting is that sometimes people will write me out of the blue and they would say, well, what do you think it should work for hearing loss? I'm like, hearing loss? How does it work for hearing loss?
Absolutely, absolutely.
And then they're like, well, hearing loss is a mitochondrial disease. And it's like, and so we tested it in a rat that's had the ears blasted for 8 hours with loud noise, and then gave the compound for a week. And sure enough, it preserved the hearing at low decibels, and the action potential from the ear to the inner brain was strong. We published on this in my review, which came out last year, called 2,4-dinitrophenol as medicine. Anyways, the point is that people then write me, well, what about, anyway, so it's really kind of fascinating how it bloomed into something, you know.
Sure, you will face your type of criticism, and they're used to it. Okay, so good luck to you.
Thank you, sir.